Thank you, Andy. Just see if I can get this up. Okay. Can you hear me okay? Okay. So I'd just like to echo the words earlier of Alison and Helen. It's really great to see so many people here to celebrate Jim's career and his legacy. It's always a wonderful opportunity to meet with old friends, and I've really enjoyed meeting up with names that I know from the past, from previous members of Jim's group. I've never previously had the pleasure of meeting in person. So I'm looking forward to chatting with some of you over coffee and then this evening at the dinner. So I thought today I would talk about polyketides, the class of molecules that I think Jim is most closely associated. And you can see here, these molecules they're structurally very diverse and they're functionally very diverse. We have many molecules on this slide that are on the WHO's list of essential medicines. And so today I'd like to not talk just about the work from my current lab, although I will touch on that towards the end of the talk, but I thought it would be nice to talk about some projects I've worked on over the last 3 decades since I joined Jim's group. So work that was done in Biotica, the company that Andy alluded to, that was founded by Jim and Peter. And before that, the project that I work on when I first arrived in Cambridge to work on these molecules. And we will be hearing about erythromycin. And so Jim worked on several of the molecules that we can see on this slide. We've heard a lovely talk about tetranacin, and we've heard about spirone. And I'll be talking about these molecules in the middle. But I just thought it was worth reflecting on these somewhat simpler looking fungal polyketides that Jim worked on in the early part of his career. So this molecule, whose name I really can't pronounce very well, I trip over it. I don't know if there's somebody, Tom, you know how to pronounce the name of this. So this was the molecule that Jim published his first paper on. It was a structure revision of this molecule. There was no NMR to help with this. This was purely done on Jim's great understanding of mechanism, of reactivity, and an incredible use of infrared and UV spectroscopy, something that today often isn't used at all. This molecule, citrulline, a fungal polyketide, an aromatic polyketide. Jim, working on these systems, was thinking about the reactivity of polybeta ketones, how you could control reactions, in thinking about biomimetic synthesis. And in that work, he generated the pentaketide precursor of this molecule in methylated form and showed that it must be that structure that's used on the route to this molecule. And out of that work, he was able to postulate the structure of the first enzyme-free intermediate, something that was contentious in the literature. Subsequently, some people disagreed with that. But just a few years ago, Russell Cox was able to show that indeed Jim was correct, using genomics and biochemistry. And this molecule also is interesting because thinking about citrulline, Jim developed a total synthesis of this molecule. And in doing that, he, I think, possibly carried out the first example of an anti-selective reaction that uses a standalone chiral base for organolithium chemistry. And then a spirone, we've heard people talk about. Actually, Finian, there was a pentaketide involved in understanding the biosynthesis of this molecule, a beautiful paper from Alison in 1995. And what's nice about the work that Jim did here was that he dissected every step along the processive pathway that leads to a spirone. It was the first example of where every step was delineated using beautiful chemical synthesis of the diketide, the triketide, the tetraketide, and the pentaketide, done in chiral form with the appropriate labels so that you could follow the biosynthesis. And it's one of these great examples of where Jim published a paper along every step of that process. So this is really groundbreaking work in these somewhat unassuming molecules. And this work was published around 1995, and it's kind of in that time period, the early 1990s, that Jim pivoted away from fungal biosynthesis to work on bacterial polyketides with Peter Ledley. And Peter had shown a few years earlier that these reduced polyketides like erythromycin are made in bacteria by these assembly line enzymes, these megasynthases. So we have along the top, we have 3 giant genes, each of which encodes for a giant protein, which has a domain organization, a protein organization of repeating modules, where each module is responsible for building the carbon chain by 2 units and to modify the structure of the beta ketone that's produced in the bond-forming reaction. And this leads to complex molecules like erythromycin. And in 1995, Jim and Peter published a seminal paper in Science, which is actually the reason I sort of joined the group, along with the fact that I'd met Sandeep in the audience here and had convinced me I should come to Cambridge and spend time. But I was really blown away by this paper because it's elegant and it's so incredibly simple. If you understand this biosynthetic pathway, Jim and Peter realized that you could rationally think about relocating enzyme domains within the assembly lines. And if you did that, for example, by relocating the thioesterase from the end of the final protein to the end of the first protein, you'd offload a molecule that would have the structure anticipated by the structure of the new hybrid enzyme. And this kicked off a whole sort of area and a slew of papers thinking about using this approach to engineer the structures of much more complex molecules. And so in 1998, they reported this paper that described the engineering of the erythromycin pathway by taking genetic information from ivermectin biosynthesis, which uses this branched chain series of starter units, these CoA thioesters here of valine and isoleucine. These are used in ivermectin biosynthesis to give you branched chains at this position. You can transfer this information, generate a hybrid enzyme that will now make erythromycins in which this structure appears in the final molecule. And it was actually this project that I got to work on when I joined Jim and Peter's lab. And when I first sort of extracted the bacterial culture and put the extract into the electrospray mass spec, which is in the basement just along the corridor here, it was literally just a series of peaks, because you don't just get these 2 molecules, you get a bit of erythromycin with the propionate starter. You have all the post-PKS engineering steps that I'll talk about in a minute. And these are reactive molecules, and you get all kinds of cyclization. So there's just a forest of peaks. And I can remember going to Jim and saying, Jim, it looks like something interesting has happened, but what I really need is an LCMS to dissect this culture extract so that we can figure out what's going on. And we were getting an instrument in the department, allegedly a few months later. It did arrive maybe 6 months later. Jim didn't want me to wait, and his response was, Barrie, we will become the LCMS. And the problem we had was that there was just one machine in the basement here. And it was really difficult to get on the machine. I don't know if you remember this, Ulf, and I was regularly fighting with members of Jeremy's group. Stuart Rowan was often on there and a great friend, but we were fighting over access to that machine. And Jim said, forget about that. Just book the machine for as long as you need. If anybody argues, send them to me. So I did. I booked it on a Saturday morning. I played it safe. And Jim said, when you do that, let me know, and I'll come down and we'll do the experiment together. So true to his word, on the Saturday morning at 8 a.m., I arrived in the room along the corridor with a pre-fractionated extract, and we spent 4 hours injecting each sample individually, processing the data, printing out the spectra, and finally figuring out what had gone on in this reaction. And I remember that was a wonderful morning. Jim's excitement was growing as we could piece together that indeed this reaction had worked. One of my favorite memories of time spent with Jim. So that understanding of modular polyketide biosynthesis led to the foundation of Bartica, a company that I spent 12 years working at. And we have some members of Bartica here today who now work at the sort of company Isomerase that followed from that. And we also have Steve Kendrew, whose work I'm going to talk about in a moment. But what's great about these systems is that they're conceptually very simple if you want to think about engineering a pathway. You have a pre-assembly phase of biosynthesis where you make the precursors. You have the assembly phase, which is that polyketide synthase that I showed you for the erythromycin or avermectin system. And then when you've released the polyketide from the enzyme, so you've got your first enzyme-free intermediate, there are tailoring enzymes that modify that structure to give you the mature product. And if you can sequence the genome of the producing organism, you can use this information in a very rational way. And in theory, you can intervene at each of these different parts of the process. You can abolish the production to make the starters, and you can feed in synthetic variants with useful handles. You can engineer that assembly line a bit like the erythromycin avermectin example that I described. And you can play around with the tailoring enzymes. You can knock genes out, you can overexpress genes, you can change their specificity. And if you put that together, you should be able to make combinatorial libraries of those complex molecules that might have improvements that might make them useful as potential valuable molecules. And we spent a lot of time at Biotica working on human medicine targets and agricultural targets. And I'd just like to tell you about one of these stories now. It's work that we did with Wyeth, which became Pfizer, on this molecule rapamycin, an iconic polyketide that's an immunosuppressive drug and used in oncology. And the group at Pfizer was sort of tasked us with the idea of can you modify this structure such that this keto group at what's called C32 is removed, because this acidic proton that sits adjacent to it drives this major route of metabolism via beta elimination. So this is the main route of metabolism, but the genes that, the proteins that are involved in this modification are expressed differentially in patients, and that leads to quite great patient variability in how that metabolism occurs, and therefore in what dosing you need to make this an effective molecule. And so the idea was here, could we make a variant that was now stable to beta elimination, but had the efficacy of rapamycin? And they knew that that would work because they'd synthesized that via a very laborious semisynthetic process. So rapamycin is a modular polyketide, and if we want to change this structure at C32, we need to change the enzyme at this specific position in the assembly line. And to do that, we would carry out an allelic replacement. We would just swap a piece of DNA in the chromosome that does the reaction we don't want for one that would do the reaction we do want. So in this case, we want to reduce this ketone group to a methylene, and to do that we have to insert the catalytic domains that would reduce, dehydrate, and then reduce again this to give you the methylene structure. So if you do that, you convert your parental polyketide synthase to your mutant. It now has this piece of hybrid DNA, and if it works, it produces our target molecule. So this is all great. In theory, we should be able to carry out some genetic engineering. We build the plasmid that allows us to do that work. We introduce it into the rapamycin-producing organism. It integrates into the genome, flips the DNA, we get the molecule. The problem with the rapamycin-producing organism is it's really recalcitrant to genetic engineering. And so Steve, who's in the audience, developed an approach where instead of using a plasmid that either had a normal origin or was a suicide vector, we used one that had a temperature-sensitive origin of replication. So this means if you raise the temperature to one which is now non-permissive, the plasmid can't replicate. If it can't replicate, it can't express its resistance gene. So if you treat with an antibiotic, you either kill the cell or the plasmid has to integrate into the chromosome such that it can now express the resistance gene. So you force the recombination. And Steve was able to show that this worked very effectively. He could select now his integrant strain where he'd integrated the plasmid into the chromosome by raising the temperature. And after he'd isolated and selected that strain, he now lowered the temperature to one which was permissive, such that in theory the plasmid would be lost from the chromosome. Hopefully it would flip that piece of DNA and we'd make our target molecule. So that's all great. Steve streaked out his colonies, hundreds of them, and we tested them for the production of this molecule. And what we got was a complete surprise. Instead of the target molecule, what we saw was a whole family of rapamycin compounds. And if you look at these structures carefully, what you see is that this backbone chain has been reduced either by 2 carbons, 4 carbons, 6, or up to 12. And what that corresponds to is the loss of the enzymatic domains for each of those steps along the assembly line. So loss of 1 module, 2 modules, or up to 6 modules. And in this case, we even see duplication of one of those modules, so we get a larger ring size. So if that's correct, we should be able to sequence the genomes of those organisms, and when we look at the genes, we should see that they now have an architecture that maps onto the loss of 1 module, 2 modules, 3, or indeed the acquisition of an additional module. And indeed, when we do that, the genotype matches the phenotype exactly. So what we've done here is go from what we thought was a very targeted experiment to a really serendipitously exciting experiment where we've diversified structural diversity of the compounds that are produced from a single experiment. We can generate libraries now of these compounds. And this turned out to be highly reproducible. Steve could do this multiple times, and he was even able to move it to a different system. So this was great in terms of a diversity generator, and some of these molecules have real potential and some other indications that we were not originally thinking about. But as we analyzed the productivity of those strains, we realized that the sites at which the recombinations had occurred, the fusion sites, tended to group in unexpected regions of the module. And the efficiency of these hybrids was really high. We were getting productivity approaching that of the wild-type organism. And that was really surprising because our best rational biosynthetic engineering methods that we'd been using prior to that, based on our best guesses at structure, a lot of that structural information coming from some of Jin's seminal ideas, was just that. It was an approximation. It was a guess at where we should be making these fusions. What we'd stumbled on here were actually fusion sites that maybe nature uses in evolving these systems naturally. And in fact, several groups have now gone on and used these types of fusion sites to carry out effective engineering experiments. And in fact, the idea of using evolutionary approaches to help guide how we would engineer these systems is really taking hold. There's a lot of people working on those approaches. So I just want to finish with a little story from my current lab, where we're interested in discovering new anti-infective molecules. And to do that, working with Matt Hutchings' group, we've been using this sort of ecology-driven approach. So the hypothesis here is that if we look in environments where microbes, bacteria that produce anti-infective molecules are important for the homeostasis of that system through certain mutualisms, we might find strains that are rich in producing new antibacterial molecules. And by looking in environments like this that have been previously undersampled and underexploited, we're hoping that we might find new structures, new antibiotic structures, which might be useful potentially for the next generation of antibiotics. So this is some work that we were doing on a plant-ant mutualism. So this is an acacia tree from Kenya. And these sort of ball structures you see are called domatia. They're swellings at the base of the thorns. And you can see here that they have these holes. And they're basically housing for fungus gardens that these Tetraponera ants are farming inside to provide a food source for their larvae. And so we were sampling in these for bacteria which might be making anti-infectives that the ants are hoping to use as sort of disinfectants within the garden, but also on their cuticles they have a range of bacteria that are producing antibiotics, again that we think are producing molecules to stop the ants bringing sort of foreign pathogens into the nest when they forage. And the reason that these domatia are produced by the acacia is because in exchange for the housing and the fungus garden, the ants act as deterrents for herbivores which prey on the acacia tree, and there's sort of anecdotal evidence that they can even deter elephants. Well, I'm not sure how true that is. Certainly they've been shown to deter sort of giraffes and other herbivores. So to cut through a lot of work, we discovered a new strain that we called Streptomyces formicae, coming from formica, from ant. And this is actually a new carbon skeleton. Two-dimensionally, it looks closely related to these actually previously known molecules, the phasamycins. These are produced by a whole array of strains. They're relatively common in nature, whereas we've only identified these so far from a single strain. And both of these classes of molecules are potent antibacterials, active against drug-resistant strains. And the phasamycins are actually biosynthetic precursors of the formicomycins. They're polyketides. They're made by a simply sort of well-understood pathway. But how you get from the phasamycins to the formicomycins was unknown. And this was really interesting for us because if you look at this ring here, you see that we've now gone from an aromatic system to a non-aromatic system and a very controlled stereospecific substitution of these two bridgehead positions. And in 3 dimensions, these molecules are very different. We've gone from an almost flat linear phasamycin structure to this twisted chair-like structure in the formicomycins. And these are actually more potent antibacterials than the phasamycins. So using a bit of bioinformatics, we've been able to show there are 2 genes encoded in the genome that code for flavin-dependent enzymes that catalyze that transformation. And these appear to have been acquired by horizontal gene transfer from a different genus of actinobacteria. And so in the first step, there's a flavin-dependent monooxygenase that appears to carry out a Biovilliger reaction. And for this, we get a lactone product that you see here. And we can propose 2 different mechanisms in which we use the flavin peroxy cofactor in either a nucleophilic or an electrophilic sense. We prefer this mechanism for various reasons we don't really have time to go into, but happy to chat if anybody's interested. And then in the second step, this lactone undergoes, we predict a hydride-initiated ring contraction. So you deliver hydride, as shown here. You get a Favorsky-like process to give you this epoxide intermediate that ultimately gives you the final phasamycin. This is based on a series of work from gene knockouts, compound purification, cross-feeding, and some biomimetic synthesis. And we've been trying to purify these proteins since 2019, and completely failing until very recently, where we've got them to be produced in nice soluble form from a Streptomyces expression platform that we've been developing based on some old work from Mervin's Bibbs lab at the John Innes Centre. So just my final slide, we're excited about these molecules, not just because of the biosynthesis, but because of their antibacterial activity. And we think these are actually dual targeting antibiotics. And that came about because we were trying to emulate some work from Sean Brady's lab, who'd shown that they were inhibitors of fatty acid biosynthesis. Really nice work, and I think some biochemical studies, some in vivo studies, comprehensively showed that these are inhibitors of FABF. But when we tried to select for resistant mutants, or we tried to overexpress the target FABF, we couldn't really see an effect, and we certainly couldn't recapitulate what Sean's group were showing. And actually, through a whole range of experimental formats, we have not been able to generate a resistant mutant to these compounds. And it turns out that's because they target a second clinically validated metabolic pathway. They target both fatty acid biosynthesis and type 2 topoisomerases. And these are the targets of clinical antibiotics such as ciprofloxacin. So we're targeting 2 metabolic pathways very potently. And unsurprisingly, when we look at the activity of these compounds against drug-resistant isolates of E. coli, resistant to ciprofloxacin, we see no cross-resistance. And this kind of dual targeting approach would explain why we don't see the selection of or the ability to select for resistant isolates. So understanding the mechanism for this is some work that's currently ongoing in the lab. We think actually it's a completely new mode of binding to these type 2 topoisomerases. So I'll finish with a few acknowledgements. Thanks to Peter, all and all of the members of his group and Jim's group that I worked with on erythromycin and different projects over the years. To the Biotica team, in particular Steve, who did all of that work, really drove all of that work on the rapamycin project. My lab currently at the John Innes Centre and collaborators. But of course, to Jim, who 29 years ago when I came in 1995, set me on course to work on polyketides, sort of my lifelong passion as well. So thank you very much.
Thank you, Barrie, for your exposition on biosynthetic engineering to work on material. Really interesting. Now,